Okay, hello everyone. Welcome to the fourth lecture of MD 6028. Last lecture we discussed viral genome expression. So we looked in detail at all the different ways the different viruses express their genome. Today we're going to look at genome replication. So we're going to look at all the different ways viruses replicate their genome. These two topics go hand in hand. So as you'll see when we work through the lecture, genome expression and genome replication are inherently linked. They're very tightly linked. We're looking at them one by one. So we're going genome expression, then genome replication, just because it would be an overload of information to look at them both at the same time. But straight away, going into lecture, I want to say that we need to bear in mind that these processes in an infected cell are happening concurrently. They're happening at the same time. So the first thing I want to do is establish what we mean by genome replication, establish some of the key principles that underpin genome replication. To do that, I want to compare genome replication to genome expression, consider how the process is different and how they're the same, because I think by now we have a good understanding of genome expression, so that's a good kind of baseline for us to start the lecture from. So this diagram here is the diagram we looked at last week, and it's a representation of genome expression in a host cell. So in a host cell, there's a double-stranded DNA genome. This double-stranded DNA genome is transcribed into RNA, and this RNA is then translated into protein. This should be a very familiar process. It's just genome expression. You've been covering it since level 4. Last lecture, however, we went into viral genome expression, which is a lot more of a complicated process. The translation part, as you remember, is the same. So viral RNA can be translated into protein, and this is how viral proteins are produced. However, the way viral RNA is produced is different to host cell transcription. Not always different, but it's often different. And the reason for that is because a lot of viruses don't have double-stranded DNA genomes. So when we were talking about genome expression last week, the main focus of the lecture really was how viruses produce RNA from their genomes. There are lots of different ways that this is done, and that's because there's lots of different viral genomes that can exist. We can have single-stranded RNA genomes. Those single-stranded genomes can be positive or negative. We can have double-stranded RNA genomes. We can have single-stranded DNA genomes. So all these different types of viral genomes that all need to produce RNA, which can then be translated into protein. And that's genome expression. Obviously, that's a very quick run through, but hopefully it sounds familiar. Next, genome replication. Host genome replication is represented here. And what you'll see is it's obviously, as I'm sure you know, a different process to genome expression. So again, in host genome expression, we have a DNA template, that's the DNA genome. This is used to produce RNA, and then this RNA is translated into protein. Host genome expression, however, completely different. We're going from a DNA genome, which is double-stranded, to another DNA genome, which is also double-stranded. So you're essentially getting replication, as the name implies. The genome is just being replicated. There's no RNA involved. There's no protein produced. It's just going from a DNA genome to a copy of that DNA genome. And one way in which we can define these two processes is we can say these are the two genomic processes that are essential for cell survival and replication. So again, we're talking about a host cell here. If a host cell wants to maintain itself, if it wants to survive, it needs to express its genome because it needs to produce the proteins that are necessary to keep a cell going, to keep a cell surviving. If it wants to replicate, so produce copies of itself. It also needs to create proteins. It needs to create these fundamental building blocks of cells that are going to be so useful, but it also needs to create copies of its genome. So when we have cell replication, we need the original genome to stay in one cell, and then we need a copy of that genome to go into the other cell. That's how replication works. If you don't have genome copies produced, then when a cell replicates, one of the resultant cells is going to be absent of a genome, so it's not going to be a functional cell. So these are the two fundamental principal genomic processes that a cell must undertake. So next we want to think about the specific enzymes that are involved in these processes. We talked in quite a bit of detail about RNA polymerases last week. And the reason why we talked about these RNA polymerases is because the RNA polymerases are the enzymes that produce RNA. So that's why they're called RNA polymerases. They're polymerizing ribonucleotides to produce RNA. We talked about the idea of template dependency. Again, if you don't know what I mean by that, if you go back to the lecture slides, the enzyme that catalyzes the production of RNA in our host cells during transcription is a DNA-dependent RNA polymerase. So that means it's dependent upon the DNA template. So it uses the DNA template, which makes sense because we have a DNA genome, and it produces RNA from that DNA template. So again, DNA-dependent RNA polymerase. For genome expression in the host cell, we understandably need a different enzyme to catalyze the process. So we're not using a DNA template to produce RNA. So a DNA-dependent RNA polymerase isn't appropriate. It's not going to work. We're using a DNA template to produce a DNA copy. So we're going from DNA to DNA. The enzyme that therefore mediates this is a DNA-dependent DNA polymerase, and hopefully that makes sense. Again, the name is just literally a description of the specific process it catalyzes. It is using a DNA template, so it's DNA dependent, and it's a DNA polymerase, which means it's polymerizing DNA. It's producing DNA from the DNA template. This is where we need to take just a slight aside to make sure we actually understand what we mean by genome replication. So we probably understand genome replication to mean making copies of the genome, and that's right, that's fundamentally right. However, we need to define what we mean by copy, and what we mean by copy is a copy that is in exactly the same format. So you can't get an RNA copy of a DNA genome. That's not the type of copy we're talking about here. You can't get a DNA copy of an RNA genome. That's not the type of copy we're talking about here. We're talking about a like-for-like copy. It has to be identical except for mutations, obviously, but it has to be in the same format. So if you have a DNA genome, genome replication must produce a DNA copy of that genome. If we have an RNA genome, and again, obviously, human cells would never have an RNA genome, but I'm thinking about viruses now. If there's an RNA genome, then genome replication must produce an RNA copy of that genome. If the genome is positive single-stranded RNA, the genome copy produced through genome replication must be exactly the same format. It must be positive single-stranded RNA. That's a real fundamental principle at the core of genome replication. Genome replication has to produce a copy that is in the same format. We'll see why this is really important later. You can see I've highlighted at the bottom this, not all viruses have double-stranded DNA genomes. And again, I'm just kind of preempting what I'm going to say later in the lecture, which I'm sure you'll be expecting. And that is that a lot of viruses can't use the same enzyme that host cells use. to replicate their genome. So we just defined host cells as using a DNA-dependent DNA polymerase to replicate their genome, and that's because they have a DNA genome, so they need to produce a DNA copy of that genome to replicate. Viruses, as we've established, don't all have double-stranded DNA genomes. Their genomes can come in all shapes, all different forms. So therefore, many viruses can't use DNA-dependent DNA polymerase. They have to use different methods to replicate their genome. They have to use different enzymes. So because some viruses do use DNA polymerase, we need to kind of establish what DNA polymerase is. So of course, not all viruses use DNA polymerase, and not all viruses have DNA genomes. However, some do. So again, I just want to establish what it is. We spent some time talking about RNA polymerase last lecture. So I just want to compare DNA polymerase to RNA polymerase. You'll actually see there's quite a lot of similarities between the two enzymes. They're fundamentally different because they produce something different. RNA polymerase produces RNA, DNA polymerase produces DNA. However, RNA and DNA are both nucleic acids. They have some fundamental similarities that mean RNA polymerase and DNA polymerase also have some similarities. So if we looked at if we look at the information from last week in which we defined RNA polymerase, we said RNA polymerase is a polymerase, poly meaning many. So RNA polymerase is producing RNA by adding subunits one after the other. And RNA is just a chain of these subunits, and these subunits are called ribonucleotides. Now, if we look at DNA polymerase, we'll see the process is, well, while the details are different, the overall process is kind of the same. And what I mean by that is DNA polymerase is a polymerase, so it is adding subunits one after the other. It's making a chain of subunits, and these subunits are composed not of ribonucleotides but of deoxyribonucleotides, and that is why DNA polymerase is producing DNA whilst RNA polymerase is producing RNA. Again, I'm sure you covered this in earlier modules. This is just a reminder, and I just want to make sure these definitions are kind of firmly established. Okay, so what we're going to do now is look through the individual groups of viruses. We're going to work through the Baltimore groups that we discussed in earlier lectures, and we're going to look at how members of these different groups replicate their genomes. Because DNA viruses have a DNA genome, so a genome that is in the same form as the host cell genome, they can, in theory, use host cell machinery, host cell enzymes to replicate that genome. So for the host cell, genome replication means going from a DNA template to a DNA copy of that template. For a DNA virus, genome replication means exactly the same. It means going from a DNA template, the genome, to a DNA copy of that template. So therefore, in theory, the viruses that have DNA genomes should be able to use the host cell DNA-dependent DNA polymerase for replication. There are also accessory enzymes. So when we're talking about genome expression and genome replication, we're focusing on the main enzyme, and that is either the RNA polymerase or the DNA polymerase, depending on which one we're talking about. However, there's other enzymes as well, which we can kind of refer to as accessory enzymes. So these are enzymes that are involved in the process, but they're not the core enzyme. They're not the fundamental polymerase. These other enzymes are still important, however, and for DNA viruses, because the process of genome replication is, again, potentially essentially identical to host cell genome replication, they can in theory use both the DNA-dependent DNA polymerase that is core to the process and also use the other host cell enzymes that are complementary to the process, these accessory enzymes that are helping the process along. And to a certain extent, we do see this. We see some DNA viruses will use a lot of the host cell enzymes, so a large number of the host cell enzymes during genome replication. However, others, despite theoretically being able to use the host cell enzymes, will actually use their own enzymes. So they will actually encode their own enzymes for various reasons. So it would be really simple and really nice if we could say all DNA viruses use all the host genome replication enzymes and machinery because the processes are the same. You're going from DNA to DNA. However, in reality, that's not the case. We see differences amongst the DNA viruses. And again, some DNA viruses will use a lot of the host cell machinery, will use a large number of the host cell enzymes. However, others will mostly encode their own enzymes to be involved in genome replication. The difference seems to be amongst this division of small viruses versus large viruses. So we can see the small DNA viruses rely largely upon the host cell genome replication enzymes. The large DNA viruses, however, don't rely so heavily upon the host cell genome replication enzymes. These large DNA viruses tend to encode their own. So most of the enzymes that are used by these large DNA viruses in genome replication are encoded in the viral genome. For small DNA viruses, again, these only encode minimal elements of the genome replication machinery within their own genome. They rely mostly upon the host cell enzymes. It is crucial to note, however, that even the large DNA viruses, so even the DNA viruses that encode most of the machinery themselves, they still rely upon some elements that are present in the cell. They still rely upon some of the host cell genome replication machinery. The reason I'm emphasizing the fact that even the large DNA viruses require some element of the host genome replication machinery, so they require at least one of the enzymes that is used by the host, is because we know that viruses, all viruses, are obligate parasites, and that means they have to infect a cell. They have to be inside an intracellular environment to complete the processes of life. And this genome replication is one of the processes of life. So one of the ways we define viruses is their necessity to utilize host cell machinery. So their necessity to access host cell enzymes and other things that to replicate their genome. So the only way they can replicate their genome is to access these host resources. And that's why this distinction is important, because if we had a large DNA virus that didn't need any of the cell's enzymes, that didn't need anything from the cell to replicate its genome, then we're stepping into a bit of a grey area about whether we can define it as a virus anymore, because we've established that one of the defining features of a virus is that it needs host cell machinery to conduct a bunch of processes, and one of those processes is genome replication. So it's not surprising, therefore, that we see all viruses, again, we're talking about DNA viruses now, so all DNA viruses, whether they're small or large, require at least one enzyme from the cell to replicate their genome. Not surprising at all, because it's a defining feature of the viruses. Okay, so we'll just go into a little bit more detail about the large DNA viruses and then also the small DNA viruses. The large DNA viruses, and this is key, they all encode their own DNA-dependent DNA polymerase. So DNA-dependent DNA polymerase is the key enzyme that we were talking about. It's the main enzyme that produces the DNA copy of the DNA genome. This enzyme is already present in the host cell. However, large DNA viruses produce their own version of it. So they produce their own DNA-dependent DNA polymerase. However, despite the fact they produce their own version of this critical enzyme, they still rely on other host enzymes to complement the process, to play some role in the genome reproduction. Example of these are transcriptional activators and topoisomerase. These are, again, we don't need to know specifically what they do. Topoisomerase, again, it's an accessory enzyme. It's kind of putting the DNA in the right position. It's allowing for unwinding of the DNA. The key thing we need to remember is that these enzymes are involved in the process of genome replication for these DNA genomes, and they are cellular enzymes. They are not encoded by the virus. The small DNA viruses, by contrast, they don't encode their own DNA-dependent DNA polymerase, so they rely upon the host DNA-dependent DNA polymerase. Again, that's fine, it makes sense because these viruses have a DNA genome like the host, and to replicate that genome they need to make a DNA copy, so they're using a DNA-dependent DNA polymerase, which is the same enzyme the host is using. The simplest way to think of this is the small DNA viruses are more simplistic, they're more minimalistic, so they rely upon the host DNA-dependent DNA polymerase because they don't have their own, whereas the large DNA viruses, like the adenoviruses we looked at in the last slide, these are large, these are a little bit more self-sufficient, so they do carry their own DNA-dependent DNA polymerase. For these small DNA viruses, and again the example here is polyomaviridae, even though they're dependent upon the host DNA-dependent DNA polymerase, they do encode at least one of their own enzymes involved in the process. Again, we talked about these accessory enzymes, these enzymes that play a role in DNA replication, but they're not the key protein, they're not the actual DNA polymerase. An example is the ATP-dependent helicase that is encoded by the small DNA viruses. So again, these small viruses, these small DNA viruses, they have to encode at least one element that is involved in genome replication. They have to encode at least one enzyme. We always see that. In the example of polyomaviruses, it happens to be an ATP-dependent helicase, which is not critical, or we shouldn't say it is necessary for genome replication, so it is needed, but it's not considered the key enzyme. It's not the DNA-dependent DNA polymerase that is actually polymerizing the DNA. It's just an enzyme that is playing a role in the overall process. Hopefully that's the DNA viruses covered in a clear way. They're the simple ones really. They're simple because, as we've said, same type of genome as a host cell. When we get into the RNA viruses now, this is where things get a little bit more complicated because, as we've talked about, host cells don't have an RNA genome, so these RNA viruses have to evolve some quite interesting and unusual ways to replicate their genome. They can't simply use host cell machinery. The host cell machinery, again, it isn't designed to replicate an RNA genome, it's designed to replicate the host's DNA genome. So first off, we're going to talk about the positive sense single-stranded RNA viruses. Just a reminder, this is what we're talking about when we're talking about RNA viruses, particularly the single-stranded viruses. We're referring specifically to class 4 and class 5 on the Baltimore classification system, so the positive single-stranded RNA viruses and the negative single-stranded RNA viruses. Again, just a reminder, this is what we mean by positive and negative. By positive, we mean encoding. We mean it contains the direct information that can determine the amino acid sequence found in proteins. Positive RNA is therefore, it's the same as mRNA. It's RNA that can be used by host translation machinery to produce protein. So it's RNA that can be translated. Negative sense RNA is different in the sense that it corresponds to a template strand. So it doesn't directly contain the sequence of amino acids, or the information that could eventually be the sequence of amino acids after translation, because it's negative. So this negative RNA can't be translated. It doesn't contain the information. The only way you can get from negative RNA to protein, so the only way negative RNA can result in translation, is if that negative RNA is used to produce a positive sense complementary copy. This positive sense complementary copy does contain the information that can determine what order amino acids will be added to constitute a protein. This positive sense RNA can therefore be translated into protein. Again, same information, just clarifying what we talked about last lecture. This is host genome expression on the left. We have double-stranded DNA, then we have positive stranded RNA, positive sense single-stranded RNA, and this positive sense RNA contains the information that is in the encoding strand of the DNA. That's why the RNA is positive sense. It contains the direct information that will determine the amino acid sequence of the protein once the RNA strand is translated. And as you can see, because it's a positive strand of RNA, because it directly contains that information, it can be translated into protein. If any of the things I've covered in the last few slides don't make sense, so if this idea of positive sense RNA and negative sense RNA doesn't make sense, it's been covered in last lecture and also the lecture before. So you just need to go back, watch the YouTube videos, or watch the slides with the audio, whatever's useful to you, and just make sure you understand those concepts. That hopefully they're well explained. If you don't find them well explained, they are also explained in the textbooks for the course. So you really want to get to grips with those concepts. Now we're going to look specifically at genome replication, which again is the topic of the lecture, and we're going to look at it specifically in positive sense single-stranded RNA viruses, so group 4 on the Baltimore classification. Here we have a simplified version of the genome expression we looked at last week. Just to run over it quickly, you can see on the left hand side of the diagram we have the positive sense single-stranded RNA genome of this particular virus, and we can see it is being translated directly into protein. There's an arrow from this viral genome directly to protein. The reason why this is happening is because the positive sense single-stranded RNA genome of the virus is essentially mRNA. It's the same as mRNA. It's indistinguishable at a cellular level by this translation machinery. So that means the translation machinery will translate positive sense single-stranded RNA into protein. Straight away, therefore, as soon as a virus with this genome enters into a cell, the viral genome will begin to be translated into protein. There's an issue here, however, in the sense that there's a limit on this process. So if this is the only genome expression that's going on in terms of the viral genome, there's a limit, because the virus comes into the cell, it comes in with its genome, and that's a finite amount That's a finite amount of genetic information, so there's only so much translation that can occur. If a virus wants to have long-term expression of viral proteins, if it wants to increase the amount of viral protein being made, it needs to produce copies of the viral genome. It needs to make more and more positive sense RNA copies, and these additional copies can then also be translated into protein. And this is what we see happening on the right-hand side of the diagram. So we go from a positive sense single-stranded RNA to a negative sense single-stranded RNA, and then we go down to another positive sense single-stranded RNA, and this positive sense single-stranded RNA is then translated into protein. The idea here, as I kind of just alluded to, is the virus is making more and more positive single-stranded copies of the genome, because these positive single-stranded copies can be translated into protein. The virus is therefore ensuring more viral proteins are being made, enabling replication of the virus. Again, these proteins, a lot of them will be going to produce new virus particles. There'll be structural proteins, for example, that compose the capsid of viral progeny. So this is why it's important for the virus to make large numbers of these proteins. The question here is, why is this pathway so convoluted? Why does the virus have to go from a positive genome to a negative genome to a positive genome again to produce protein? And the reason for this is, again, something we talked about last week. It's this idea that if you have a positive sense single-stranded RNA genome, you can't make another positive sense copy of that without using a negative sense intermediate. So the way the RNA polymerases that are making these RNAs work is, if you have a single strand of RNA here, the RNA polymerase will progress along that single strand and add a complementary strand. Okay, so that means if you have a positive strand of RNA, an RNA polymerase will add a negative strand here. Therefore, if you're using a positive sense single-stranded RNA, you can only produce a negative strand from it. Similarly, if you then have a negative strand, you can only produce a positive strand from that, because the RNA polymerase is adding the complementary strand. So that's why this kind of seemingly convoluted mechanism exists, because the virus is starting off with a positive sense strand, and it is using that as a template to produce a negative strand, and it is using the negative strand as a template to produce positive strand. And these positive strands that are produced can then be translated into protein. And it's clear to see how, if you have an initial small amount of RNA, so if you have a fixed amount of genome coming into the cell within the virus, which you do have, this fixed amount can be used to make lots and lots of negative strands. So there's no reason why you can't have a single positive strand being used to produce more and more and more and more copies of negative strands. It's just the RNA polymerase going over and over and over again. Then all these negative strands can be used to produce many positive strands, and these positive strands can be translated into protein. And in this way, a process that starts with a single copy of the viral genome can end with a large amount of protein being produced. So hopefully that makes sense. Interestingly, for a lot of positive sense RNA viruses, the process of genome replication is exactly the same as the process of genome expression that we just looked at. And it should be clear why when we walk through the process now. So we're starting off with this positive sense single-stranded RNA, as we said. If we want to replicate this genome, we need a copy of the genome that is exactly the same, that's in the same template. Remember, the key principle of genome replication is, if you start off with a genome in one form, you have to end with a copy in the same form. If the cellular genome is double-stranded DNA, which it is, for that genome to be successfully replicated, you need to end with a double-stranded DNA copy. Can't have an RNA copy of a DNA genome, can't have a single-stranded copy of a double-stranded genome, has to be the same. Otherwise, it's not replication, right? So for these positive sense single-stranded RNA viruses, genome replication must result in the production of a positive sense single-stranded RNA copy. Well, the process we've just looked at of genome expression, that's exactly what it produces. Genome expression involves a negative sense single strand of RNA being produced from the positive sense single-stranded RNA genome, and then involves a positive sense single-stranded RNA being produced from the negative sense single-stranded intermediate. All we're doing is taking away the actual translation. So we're just taking away the end point of the process. So we have this positive sense single-stranded RNA produced. In genome expression, this strand would be translated into protein. However, in genome replication, this strand isn't translated into protein. It's retained and it's packaged into new virus particles, and it is their genome. It serves as the virus genome for each of these viral particles, because again, it's exactly the same as the virus genome that came into the original cell. It's positive sense, it's single-stranded RNA, it's a copy. A good example of a virus that replicates its genome in this way is poliovirus. So poliovirus is a positive sense single-stranded RNA virus. It's a good example because it's quite clear. It's pretty clear cut. Also, we talk about it in later lectures as being a virus that causes human disease. It replicates in motor neurons and causes paralysis. So it's an interesting virus. And this is the genome replication process here. And you'll see it's familiar. It's just the process we looked at in the last slide. Poliovirus has a positive sense single-stranded RNA genome. This positive sense single-stranded RNA genome is used to produce a negative sense single-stranded RNA copy. This negative sense single-stranded RNA copy is then used to produce a positive sense single-stranded RNA copy. This positive sense single-stranded RNA copy is the same as the original genome. It's the same as the positive sense single-stranded RNA genome that entered into the cell. And this positive sense single-stranded RNA genome that's produced through this process is then packaged into the poliovirus viral particle. And within this particle, it serves as the genome for this particular virus particle. Few other examples here. I'm mentioning these because they're quite important viruses. So the flaviviruses, these include quite a few that have a significant impact on human health. So dengue fever caused by dengue virus, West Nile virus, Zika virus, hepatitis C virus, and we also have poliovirus, which is a member of Picornaviridae, which we just looked at. Okay, so we've looked at genome expression and we've looked at genome replication, and everything we've said is completely true. However, there's just a little bit more to look at. There's a little bit more detail we haven't explored yet, and this detail revolves around the switch from genome expression to genome replication. I said in the earlier slides that we can't think of these processes as completely distinct because they're not happening in different parts of the cell. They're happening in the same part of the cell. They're happening to the same genome. It's the same genome being expressed and being replicated potentially. So we need to think about how these processes are related to each other. A few slides ago, we considered how in some viruses, the positive sense single-stranded RNA viruses, the processes are the same. It's the same process, and that's absolutely fine. However, that's not always the case, and the positive sense single-stranded RNA viruses, where the processes are the same, they exist, but it's not all positive sense single-stranded RNA viruses that operate this way. And what I want to do now is look at some of the positive sense single-stranded RNA viruses in which genome expression and genome replication are different processes, and we're going to look at why they're different. Essentially, the difference is to have a switch between the two, so it enables the virus to switch from one to the other, and we'll see what I mean by that in a second. But firstly, I want to think quickly about why it might be useful to have this switch. So why is it useful for a virus to be able to go from genome expression to genome replication? And that's almost always the way it goes. It's almost always a virus starts off expressing its genome, then there's a switch, and it starts replicating its genome. The reason is it's beneficial for the virus to be expressing proteins, to be producing proteins in the early stages of infection. So when it first infects a cell, it's beneficial for the virus to be expressing its proteins. There's a couple of reasons. One reason is that some of these proteins that are made can have an active role in infection. And we looked earlier how some of these proteins, last lecture, how some of these proteins can be potentially actively involved in processing the genome. So these can be polymerases with an active role in infection. Other enzymes can be enzymes that are proteins that counteract the immune response. So these are proteins that promote infection by preventing the immune system from disrupting infection. Obviously, it's really important to have these proteins made and active as soon as possible. The other reason is the proteins that are made from the viral genome, so these proteins that are expressed, or the genes that are expressed to produce the proteins, encode proteins that are going to compose the virus capsid. So these are structural components of the virus capsid, and these proteins will be assembled to produce new virus particles, so to produce progeny. And this is ultimately the purpose of infection. So it is beneficial for the virus to start producing these structural proteins as soon as possible. It enables the virus to produce more and more of these structural proteins, which ultimately results in the creation of more and more progeny, which again allows the virus to proliferate. Ultimately, there's a switch to genome replication. This genome replication produces copies of the viral genome, and these copies can be packaged into the virus particles that are produced. This switch doesn't need to happen early because first you need the virus particles to be made. First you need the viral proteins to be created, and these viral proteins can be assembled into new virus particles. Then you need viral genome copies to be produced, and these viral genome copies can be packaged into the new virus particles. So hopefully it makes sense why many viruses start off expressing their genome, and then there is a shift where they start replicating their genome. So it goes genome expression, then a shift to genome replication. This slide represents the shift that can occur between genome expression and genome replication, and the main takeaway is this idea of subgenomic fragments. So when the genome is being expressed, previously in previous slides we've thought of the genome in its entirety being expressed. That's not the case often. Often you have subgenomic fragments being expressed, so just portions of the genome being expressed. So if we look at the system overall, we're going from positive sense single-stranded RNA to negative sense single-stranded RNA, and using this negative sense single-stranded RNA, short subgenomic fragments of positive sense single-stranded RNA are created. These positive sense single-stranded RNA fragments, again, they're just like RNA, so they're just like mRNA, so they can be translated by host translation machinery. It doesn't matter that they're subgenomic fragments, it doesn't matter that they're small portions of the entire genome. They can be translated into protein, no problem at all. And this is exactly what's happening in the early stages of viral replication. Ultimately, there is a shift, and the shift is a shift from these subgenomic fragments being produced to full genome copies being produced. Obviously, to produce progeny, full genome copies are needed. For each round of replication, you can't be losing bits of the genome, because a few genome copies later, a few rounds of replication later, and you've got no genome left. So to package into each new virus particle, a full copy of the genome is needed. And that's why when we're looking at genome replication, we're always looking at full length copies of the genome. And again, hopefully this diagram kind of demonstrates that very well. When we're talking about genome expression, we have these short genome fragments, and then we have this switch to full length genome production for genome replication. Again, not all viruses do this switch, and not all positive sense single-stranded RNA viruses do this switch. Sorry, these are the viruses we're talking about now. But a few slides ago, we talked about poliovirus, and poliovirus is a positive sense single-stranded RNA virus that doesn't do this switch. It just makes these full-length copies, and sometimes these full-length copies are translated into protein. Sometimes they're packaged into virus particles. However, there's no role for subgenomic fragments. Subgenomic fragments just aren't made in the case of poliovirus. In this example, however, they are made, and these subgenomic fragments define the genomic expression portion. Full-length fragments define the genome replication portion. Hopefully I've explained myself a little bit more here. We can see, unlike subgenomic fragments, full-length transcripts can be incorporated into new virus particles. Again, I hope I explained why it's necessary to have full-length genome copies going into each new virus particle. The reason is, if you miss any of the genome, if you have bits missing, then after just a few rounds of replication, you've got no genome left. Even in the next round of replication, the bit that's missing could be encoding a really important gene. It could be importing a key gene. So all this process is for nothing, because the progeny being produced are useless. They can't infect a new cell because they're missing a really important gene. So it's crucially important that genome replication results in the production of an exact, full genome copy. The question now becomes, how is this switch controlled? So I've told you a lot of positive sense single-stranded RNA viruses will start off expressing their genome, but then will switch to genome replication. How is this switch controlled? How does the virus go from producing these short subgenomic fragments of positive sense single-stranded RNA to producing full-length genome copies, which are composed of positive sense single-stranded RNA? We can't go into details of how every single virus that undertakes this process controls the switch. However, we can look at some specific examples, and one example we're going to look at is the example of Syndbis virus. Syndbis virus is a toga virus, meaning it belongs to the Togaviridae family. You'll see here we have the phenotypic classification system for RNA viruses, and the toga viruses are single-stranded positive sense RNA viruses. This is, again, the idea that these positive sense single-stranded RNA viruses will all start off with a positive sense single-stranded RNA genome. This positive sense single-stranded RNA genome will be used to produce a negative sense single-stranded RNA copy, and then this negative sense single-stranded RNA copy can be used to produce either positive sense single-stranded RNA in the form of subgenomic fragments that will be translated into protein. This is genome expression, or the negative sense single-stranded RNA intermediate will be used to produce positive sense single-stranded RNA full-length genome copies. And these full-length genome copies represent genome replication because they are identical to the original viral genome. And again, this is what we see for Syndbis virus. So we see the positive sense single-stranded RNA genome being used to produce a negative sense copy, then this negative sense copy being used to produce positive sense subgenomic fragments, namely 26S subgenomic mRNA, which is a particular subfragment. And this subfragment is then treated as mRNA. It's translated by the host translation machinery. There's a lot of RNA being produced from RNA here. You can see there's two stages where we have an RNA template being used to produce more RNA. So obviously we need an RNA dependent RNA polymerase to do this, or the virus needs an RNA dependent RNA polymerase, I should say. And again, if you look back at the lectures from last week and the week before, we talked a lot about this. We outlined how and why these RNA viruses need an RNA dependent RNA polymerase. And we also talked about how for these positive sense single-stranded RNA viruses, they can simply encode an RNA dependent RNA polymerase within their genome. So this RNA dependent RNA polymerase that the virus uses, it isn't brought into the cell with the virus, it's just encoded in the genome. And the viral genome is then directly translated into protein. And the protein that's made is the RNA dependent RNA polymerase. So again, if you go back to the last lecture, you'll see that the genome of these positive sense single-stranded RNA viruses can be directly translated into protein because it's positive sense RNA. It's basically mRNA, and that's what happens. The only issue with this is you eventually run out of genome. You can't maintain high levels of expression. So the way viruses maintain high levels of expression is what we're looking at in this slide, where you get more and more positive sense copies produced. So the RNA dependent RNA polymerase that is used by Syndbis virus is called P1234. And again, this protein P1234 is encoded in the viral genome. And as soon as the virus enters into the cell, the positive sense single-stranded RNA genome of the virus is translated into protein. P1234 is therefore made. It's the protein that's made. And P1234 can then act as an RNA-dependent RNA polymerase and mediate the production of more and more 21S subgenomic mRNA fragments by the mechanism we're looking at in this slide. Translation of the Syndbis virus genome as it comes into the cell also has another product, so it doesn't just result in the production of P1234, it also produces what we call NSP proteins. Now these NSP proteins, because they're being produced in this way, will build up over time. So you get this gradual increase of NSP proteins in the cell. It's reasonably slow because they're produced by translation of the original genome, not of these subgenomic mRNAs, but nonetheless what you see is a build-up of NSP proteins in the cell. Once these NSP proteins reach a certain level, these NSP proteins will process P1234, so they'll change P1234 in a way that changes its specificity. So once these NSP proteins have acted upon P1234, you'll see that P1234, which is again an RNA-dependent RNA polymerase, will change the way it acts upon the negative sense RNA strand, and you will no longer get 26S subgenomic mRNA produced from the negative sense RNA strand. You'll get 49S full-length genomic RNA produced. And this is what that looks like. We're essentially looking at a switch from the 26S subgenomic mRNA to full-length genomic RNA. Again, we're calling it 49S genomic mRNA. What it is an exact copy of the original genome. And this is the switch. This is the mechanism that mediates the switch. It's the buildup of NSP proteins. It's quite a clever mechanism actually, because it takes time for these NSP proteins to be produced. It takes time for them to build up within the cell. And as they're building up to a critical level, after which they can act upon P1234, other proteins are being produced, other products are being made because these 26S subgenomic mRNA fragments are being produced by P1234, and these subgenomic fragments are then being translated by the host cell translation machinery. Again, once we have enough NSP proteins in the cell, there's this switch to the production of 49S genomic mRNA, and this represents the switch from genome expression to genome replication, because these 49S genomic RNA copies are full-length copies. They're essentially full-length copies of the synvis virus genome that can be packaged into new virus particles. So to summarize what we've just talked about, we have two mechanisms for genome replication in these positive sense single-stranded RNA viruses. The first is really easy. It's the same as genome expression. You have positive genome that is copied into a negative copy, then this negative copy is used to produce a positive copy, and this positive copy is the genome. It's identical to the original genome, so it's packaged into new virus particles. The second method of genome replication in these positive sense single-stranded RNA viruses involves an active switch from genome expression to genome replication. An active switch from the production of subgenomic fragments, which are translated, this is genome expression, to the production of full-length genome copies that represent exact copies of the genome, essentially. This is genome replication. So again, hopefully that was clear. Now we want to consider the negative sense single-stranded RNA viruses and how they replicate their genomes. So we're talking about the fifth group in the Baltimore classification system. So negative sense single-stranded RNA viruses. So we covered genome expression for these viruses last week. The only additional information I want to provide is to say that, again, as with many of the positive sense single-stranded RNA viruses, these negative sense single-stranded RNA viruses will express their genome through genomic fragments. So it's not actually full-length positive sense transcript being produced from the negative sense genomes of these viruses, but it's subgenomic fragments being produced, and these subgenomic fragments are then translated into proteins. So if you compare this slide to the slide we looked at for the negative sense single-stranded RNA viruses last week, you'll see we're talking about exactly the same thing. We're talking about a negative sense single-stranded RNA genome being used to produce positive sense single-stranded RNA copies, and then these positive sense single-stranded RNA copies being translated into protein. The only extra bit here, the only bit that's different now, is that we're introducing the idea that we're not looking at full length copies being made, we're looking at subgenomic fragments being made. This is what genome expression actually looks like when we're looking at it in a bit more detail. Next, we need to think about genome replication. So we're introducing the idea of genome replication into the mix. And again, we're seeing that genome replication does involve the production of full length fragments. Just like with the positive sense RNA viruses, we have subgenomic fragments being used for genome expression. Then we have full length copies of the viral genome being produced for genome replication. The way this works in the negative sense single-stranded RNA viruses is the negative sense genome is used to produce a full-length single-stranded positive copy of the genome. This full-length positive copy is then used to produce a full-length negative copy. So if you remember, the final product of replication has to be the same as the initial genome. If you have a negative sense single-stranded RNA virus, then genome replication has to produce a negative sense single-stranded RNA copy of the genome. That's just one of the fundamental principles of genome replication. And that's why we have this additional step at the end, where the positive sense single-stranded RNA copy of the genome is used to produce a final negative sense single-stranded RNA copy of the genome, which represents the final product of genome replication. Now, again, we need to consider how do the viruses switch between these two mechanisms? How do the viruses switch from genome expression using these short subgenomic fragments of RNA to genome replication using full-length genome copies? To answer this, we're going to look at influenza. So influenza is a virus with a negative sense single-stranded RNA genome. So it's a great example here. We're going to look at how influenza switches from genome expression to genome replication. So what we're looking at for influenza is essentially what we just discussed. We're starting off with genome expression, which involves the production of these short subgenomic fragments. As we can see here, these subgenomic fragments are translated into protein, and that is why we're defining this process as genome expression, because it's resulting in protein being produced. So now what we want to think about is, how does this switch come about? So we're starting off with genome expression. How do we have a switch to genome replication? It all revolves around this protein called NP protein. So one of the proteins being produced via expression of the genome, so through translation of these subgenomic fragments, is called NP protein, and this NP protein is instrumental in the switch. As we get continued expression, we see NP levels rise and rise in the cell until they reach a critical level. And what we have here is a representation of exactly how NP protein mediates the switch from genome expression to genome replication. So we have continued expression for a while, and the viral proteins are being produced. And ultimately what happens is the levels of the NP protein build up in the cell, and once they reach a critical level, they begin to coat the nascent RNA being produced. So negative sense RNA genome of influenza is being used to produce positive sense RNA copies. As these positive sense RNA copies are being produced, NP protein coats them and prevents the production of subgenomic fragments. They prevent the essentially cleavage reactions that are mediating the production of subgenomic fragments. And as a result of NP protein coating the positive sense RNA transcript that is being produced, The positive sense RNA transcript is kept as a whole, so there's no subgenomic fragments produced. There's a full-length positive sense RNA copy of the genome produced that is coated in NP protein. So this is how NP protein mediates the switch from genome expression to genome replication. It does it by mediating a switch from subgenomic fragments to full-length genome copies, which is again how we distinguish between genome expression and genome replication, essentially because genome replication requires full-length genome copies to be made. Now there's just one final step, which I'm sure you predicted, because we've said that for genome replication to be complete, the product has to be in the same form as the original genome. So influenza has a negative sense RNA genome, so the final product of genome replication has to also be composed of negative sense RNA. This means that the positive sense RNA genome copy that was produced, that is coated by NP protein, and that is a full-length copy of the genome, must be used to produce a negative sense RNA copy that is also full-length, and that is now a true copy of the original genome because it's in the same form. So it's essentially exactly the same as the original genome. These negative sense copies can then be packaged into new virus particles, and that's the end of the story. And by end of the story, I mean that's the end of this round of replication, as it enables the fully formed new virus particles to leave the cell complete and to infect new cells, thus continuing the next round of replication. Next, we have another example, which is vesicular stomatitis virus. This virus is also a virus with a single-stranded negative sense RNA genome. However, it's a member of the Rhabdoviridae family. You'll see the process is the same, essentially. It's just another example. We have a negative sense single-stranded RNA genome that is being expressed in these short subgenomic fragments, and this is genome expression. A switch to genome replication is required to produce these full-length RNA fragments rather than the subgenomic fragments that are defining genome expression. So the virus wants to go from genome expression using subgenomic fragments to genome replication, which is defined by these full-length fragments being produced. And again, we see the same situation really. We see a buildup of protein products, and these protein products are being produced by genome expression, because that's what genome expression is, right? So the viral genome is expressed, and it's expressed through these subgenomic fragments. And as a consequence, there's a buildup of NP protein, which is a specific viral protein. As the levels of this NP protein build and build, NP starts to coat the nascent RNA that is being produced. As it's coating the nascent RNA that is being produced, it's encouraging the production of full-length copies rather than the subgenomic fragments that were being produced initially. Therefore, as the NP protein builds up within the cell, there is a switch from the production of these subgenomic viral fragments to the production of the full-length copies of RNA. And again, this is genome replication when the full length is being produced. And that's what we're seeing on the left here. We're seeing the full-length RNA copy of the genome is fully coated in NP protein, and again, it's full length. It's not subgenomic fragments, it's a full-length copy. The final step is this full-length copy, which is positive because the original genome is negative, so a positive copy is produced of the negative original. This positive copy then needs to be used to produce a final negative copy, and this negative copy is essentially exactly the same as the original viral genome. It can therefore be packaged into a new virus particle, and that's the process of genome replication complete. So those are the negative sense single-stranded RNA viruses. Next, we want to go to the double-stranded RNA viruses. So we've talked about all the other groups so far, and logically where we're up to now is double-stranded RNA viruses. Again, these are viruses with a double-stranded RNA genome. Once again, this is what we're thinking about when we're talking about a double-stranded RNA genome. We're talking about a genome that is positive RNA paired to negative RNA, so double-stranded genome. So we have both a positive strand and a negative strand, a template strand and an encoding strand. It looks like cellular genomics. It looks like a cellular genome, but remember it's RNA. So we do have a double strand, just like we have in the cellular genome. However, the double strand in the cellular genome is of course DNA, and what we're talking about here is RNA. Therefore, to replicate this genome or to express this genome, what is needed is an RNA-dependent RNA polymerase. So whether we're talking about replication or we're talking about genome expression, an RNA copy is needed. We're talking about producing RNA from an RNA template. It's important that we remember when we're talking about double-stranded RNA viruses, we're talking about them almost as an aside, just because they're rare. There's not that many of them. Most RNA viruses are single-stranded, and we talked about this in an earlier lecture, how RNA is most comfortable, so it's lowest energy when it's single-stranded. If you have double-stranded RNA, it's not as stable. It tends to exist as a single strand for that reason. And DNA is the flip side. DNA is the opposite. DNA is most stable when it's double-stranded. That's why most DNA viruses have double-stranded genomes. Not all. We do have single-stranded DNA viruses, but these are general rules. Most DNA viruses have double-stranded genomes, and most RNA viruses have single-stranded genomes. However, we're considering now the minority of RNA viruses that do have double-stranded genomes. Included in this group are the reoviruses, so members of the viral family Reoviridae. So just a diagram here, so we know what we're thinking about here. We have the virus with the double-stranded RNA genome in the middle. So the first step, fairly simple. So RNA copies are produced within the internalized virus particle, then exported into the cytoplasm where they're translated into protein. So there's an RNA-dependent RNA polymerase enzyme that is acting in the virus particle that is producing positive stranded copies of the RNA genome. These positive stranded copies are being exported out of the virus particle where they can be translated by host cell machinery into protein. This protein is, of course, it's viral protein. It's produced by expression of the viral genome. So the protein that's produced it's viral. It's what is encoded in the viral genome. These proteins can therefore be assembled to make new virus particles because virus particles are composed of viral protein. Within the new virus particles, we have packaged a positive sense copy of the viral RNA genome because, as we just said, the virus is exporting positive sense copies of its RNA genome into the cytoplasm where the positive sense RNA copy can be translated into protein. But some of these copies will be packaged into new virus particles. So essentially now we're talking about replication. So at a molecular level, the process is the same. It's just that some of the copies produced are being expressed into protein, translated into protein. Some are being packaged into the new virus particles. And that's what we have demonstrated here. We can see positive sense RNA copies are incorporated into the new virus particle. But the process can't be complete at this stage because we know the double-stranded RNA virus genome is double-stranded RNA. That's why we consider them double-stranded RNA viruses. So the positive RNA copy of the genome has to be converted into double-stranded RNA. And this is simply done by the same RNA-dependent RNA polymerase. So the RNA polymerase will progress along the positive strand and produce a negative strand, and they're double-stranded RNA. Nice and simple. And again, bringing this back to the specific enzymes, we need to remember we're talking about an RNA-dependent RNA polymerase. So it's an RNA polymerase because it's producing RNA, and it's RNA-dependent because it's using an RNA template. So this brings us to group 6 and group 7. These viruses express their genomes very differently to members of the other groups, and the key is the fact that they use an enzyme called reverse transcriptase. Reverse transcriptase mediates a process called reverse transcription. Reverse transcription describes the process by which an RNA template is used to produce DNA. So the enzyme reverse transcriptase can be defined as an RNA-dependent DNA polymerase, and we went into that in quite a bit of detail last week, so I hope that's clear. This week, so the genome replication of these viruses is very interesting. Again, it's distinct from the other groups. We're going to talk about it, but I want to talk about it slightly separately to this lecture. So what I want to do is talk about retroviruses on their own in a little bit. So we are going to discuss genome expression again and genome replication for these members of group 6 and group 7, but not right now. I think this lecture is packed with information. I don't want any more technical information in it really, so we're just going to look at that a little bit further on. So for now, don't worry about genome replication for these groups. We will study it and we'll give it the time it needs. I just think there's enough in this lecture, to be honest. Yep. So now what I want to talk about is genome structures. So I just want to give a few slides discussing the different structures that viral genomes can have. We've kind of been thinking of viral genomes as coming into the cell mostly as linear. That's often true, but it's not always true. There's sometimes little differences across the different genomes, so little structural differences. What I want to do is clarify those differences and just make sure we're aware of them. So first, we want to think about what we call segmented genomes. So we tend to think of the genome of each virus as being one strand that's either single-stranded or double-stranded, but it's all together. It's one piece of genetic information. However, often the genomes are what we call segmented, so they'll be composed of multiple pieces, as shown in the diagram here of Virology. So this is the double-stranded RNA virus family we were talking about, and you'll see that the genome of the viruses, members of this family, is split into these segments. We're not talking about subgenomic fragments here. The shorter fragments aren't copies of the bigger fragments. They're distinct from each other, and together they compose the genome. So this virus has a full genome. The genome just happens to be in multiple segments. And I know a few slides ago I was showing you this genome as one piece, as I am in the diagram on the left here. However, that was just a simplified version to make the point I was making. In reality, we see that these double-stranded RNA viruses, like some other viruses, have segmented genomes. So they have a genome that is split into separate pieces, and these genomes together, so these segments together, create the full genome. So here we have another example of a segmented genome. We have an influenza virus on the right, and you can see its genome isn't one distinct structure. It's actually a number of different segments. The interesting thing here is that each of these segments are not linear, they're looped, so they attach at the ends, and that's what we see for influenza. We see these multiple loops, and each loop is a part of the genome. Contrasting this, we have HIV on the left, and you see we just have 2 copies of the genome here. These aren't segments, they're just 2 copies. So HIV is an example of a virus genome that isn't segmented. Influenza is a good example of a virus genome that is segmented, just like the reoviruses we looked at a slide before. Okay, next up we have the double-stranded linear and single-stranded linear genomes. These are the ones we're familiar with. These are the ones that all the diagrams we've looked at tend to show. However, some viruses, again, don't conform to this structure. An example here is of a circular genome. So it's important to note that everything we've talked about so far is absolutely right. Everything applies. Just because these viruses have genomes in a slightly different conformation, that doesn't mean they express their genomes in a different way to what we've talked about. A double-stranded DNA virus genome is still a double-stranded DNA virus genome, whether it's linear or whether it's circularized. The only difference is it's joined at the end. So it's really not super important information in terms of everything we've talked about in the last few lectures, but it's something we need to know about. We don't really want to have a vision in our head of genomes that are circular as being linear, because it's just not correct. So we just need to bear in mind that some viral genomes can circularize, so the ends can attach, and that's what we see here. Finally, we have the gapped genomes. So some of these DNA viruses that have circularized genomes also have gapped genomes. So a circularized genome that has a portion missing from one of the strands is considered gapped because there's this gap in the genome. We actually talked about this recently. We were looking at genomic processes of hepadnaviruses, and hepadnaviruses are one of the viruses that have a gapped genome, so this should be familiar to you. As with the other types of DNA virus genome structure we've just looked at, the normal principles apply. Everything we've talked about in the last couple of lectures of how DNA viruses express and replicate their genomes completely applies. It's just we need to be aware of the fact that some of these genomes are circular or circular with a gap. Okay, thank you everyone. Thank you for watching this recording. I hope you found it useful. I hope it was interesting, and I hope everything was clear. If anything isn't clear, you'll have the opportunity to discuss it and ask any questions you have in the tutorial this week. So thanks again, and I'll see you there.